I'm going to talk about the future perspectives of TDM in the community, because we've heard a lot about the hospital, but yeah, what's happening with our patients when they are discharged? So we've heard a lot about better technologies, advanced technologies, so maybe these are suitable for patients at home. So point of care tests, maybe something of interest. And also whether this can be a new service that pharmacists can deliver to our patients. And so we all know this picture, that there's lots of pharmacokinetic variabilities caused by malabsorption, comorbidities, drug interactions, toxicity, renal function loss. And of course, that doesn't happen only to patients in the hospital, but also patients at home. So if patient at home needs TDM, typically a GP or a specialist would provide the patient with a form, and then patient needs to go to a pathology service or the hospital, have that sample collected. Then that sample needs to be transported to a laboratory. And then of course it needs to sit in the freezer and wait until it is analyzed. And often that is done in batches, especially with chromatographic assays. When results are ready, they need to be communicated to the physician, and depending on where that physician is, that maybe go by mail or by phone or through the medical record system. And of course, when the physician has seen the result, it needs to be, sorry. Let's go back. I think everybody understood what I talked about here, about all the factors influencing pharmacokinetics. So we were going to the whole, let's say, chain of actions that needed to be performed for a patient to receive TDM in an outpatient setting. And my conclusion was that it is a long turnaround time before you finally can adjust your dose. And of course, in a home setting, you can make use of alternative sampling techniques, which is practical, but it does not really speed up the process per se. So when we think about point of care testing, and we talk about testing that are performed near or at the site of a patient, with the result leading to possible change in care of that particular patient. And so we have a few of them, like looking at your blood clotting or your blood glucose. We have now, I think most of us have used one of these rapid COVID tests, but also tests for drugs of abuse make use of these types of analysis, showing a positive or a negative result. But of course, for TDM, a positive or a negative result is not very helpful because you want to tailor your dose. So we have nothing yet for our drugs that we need to adjust. So the challenges are that there's really no data out there. The question is there a commercial interest? Because TDM typically is not a highly commercial interesting area. And then we're talking about home situations, potentially with fewer patients. At this moment, we don't have dedicated equipment yet. And of course, reimbursement is important. So there are many issues that we need to solve before we can make this happen. So when I started looking at saliva as an easy sample to collect in a home situation or in outpatient setting, I came across this paper from 1978 from Meiner Danhoff and Douwe Breimer, two famous Dutch pharmacologists, and they produced a very nice review, producing all the information that was available at that time on the correlations between saliva and plasmas. And there you can see on this slide where the phenytoin concentration in saliva is compared to the plasma-free phenytoin concentration, that there was a very nice correlation. And that indeed they said that for some of these drugs, there's enough data to move towards saliva TDM, but for other drugs, there seem to be some issues that need to be resolved first. So when we look at saliva and how it is produced, it's produced in these glands, and the actual saliva is hypotonic compared to plasma. The pH range can vary a little bit, and of course that may have some influence on how drugs behave under that pH condition. And of course, there are many factors that influence how much saliva is produced. You can imagine if you're really dehydrated, that saliva production is low, while if you're looking at the buffet, as we did this evening, your saliva starts flowing. So indeed, there is influence of, let's say, environmental factors on how your saliva is produced. And then, of course, the behavior of the drugs in saliva also varies. So there are limitations of using saliva. And then we found that many of the studies done are in single dose studies. And the behavior in the single dose study may differ a little bit from dosing at steady state. Especially absorption phase are difficult to assess. Now, of course, we've seen that not all drugs penetrate in saliva. And this is a nice example of one of the studies we did on amikacin, where we did full PK curves. And the saliva was a nice flat line. Well, we had a proper concentration time profile for the serum concentrations of amikacin. And of course, as mentioned, variation in saliva flow. How do we interpret that in relation to the concentration? And of course, if we take our drugs orally, what does reside in our mouth and may affect our saliva concentration? So rinsing your mouth before collecting a sample. Ideally, you collect samples pre-dose and not post-dose. But these are all need to be taken into account. But still, the analysis requires one of these fancy machines, HPLC, LCMS, GC. And of course, in home situations, you still have to send your sample to a laboratory, wait for a result. So this is not really helpful. So although it's patient-friendly, we get some additional challenges in interpretation, and the analytical pathway at this moment is still the same. However, if we look closely at UV, there may be some bright light in the future. And you know, saliva is a clear solution, and if we look at the background behind UV spectrum and the use of concentrations and absorptions, there may be something that we can help to facilitate TDM in outpatient setting. And that is these type of NAFT machines. And we know UV spectrophotometers traditionally were huge and sitting on the benchtop. But nowadays we have these very small portable devices, often with a battery pack. And these can definitely be used in an outpatient setting. Now we are using the NP80 from Implan. And I've stated here my conflict of interest, that it was purchased by a grant from the City Pharmacy School. And I have no stock or shares in this company. But you can see other companies produce similar type of equipment that likely do the same job. So when we're talking a little bit about the vision of TDM in community pharmacy, we see our patient, and typically in Australia, you go monthly to your pharmacy for your new scripts. And at that time, the pharmacist can find out that you have a drug that may be supported by TDM. As saliva collection is discussed, there is some situations when you're facing a patient with a bacterial infection that you may want to sterilize the saliva sample before further analysis. Then, of course, you have your analysis, you have your interpretation of your results, either through a standard dosing algorithm or using fancy Bayesian software, but then we have a result. And as we have pharmacists present, they can interpret those results and immediately phone the GP or the consultant and make appropriate changes where needed. So for saliva collection, often these salivettes are used, and then in that picture you see on number 6, the centrifuge. So that's an additional device that you would need to use these samples. However, alternatively, you can just put that cotton roll in a syringe and just push the plunger and squeeze the saliva out of the cotton roll. And that's what we did in several TB studies. Worked quite well without the need of a centrifuge. And we can also use the saliva deposit type of containers, we all know from COVID. So there are alternative low equipment tech procedures to collect that saliva. And of course, we are worried about contamination, especially if we're talking about microsampling, saliva sampling for patients with infections. So we did kind of a test. We used a culture with lots of bacteria, and then we use these microfilters. They're well known in our sterile suite to make sure that our IV preparations are sterile. And we just filtered the solution through the filter, and then we cultured the results coming from that filter, and it was all still sterile. And so we were able to get rid of all the TB bacteria by just simply using these small filters. So when we look at least our machine in a little bit more detail, there are two types of sensors on this platform. So there's the nano sensor, where you can just put a small drop of the fluid, or you can make use of the cuvette method, where you have a larger sample, but it also allows you to measure more sensitive. There's even a mini vortex on the platform on board to make sure that your sample is homogeneous. And then, of course, with all these modern touch screens, you can have your results, interpret the results, have background filtration, and store your samples and the data. So when we're looking at sample measurements, and I will now talk you through this example of levofloxacin, you can see here on the first graph different concentrations from 40 milligrams per liter, 20, 10, 5, and 2.5. You see the different shades of gray. But the biggest peak in this spectrum is caused by amylase in saliva. So that is actually something you should be careful about. And this was actually the levofloxacin peak. So we enlarge it a little bit, and you see there's a clear difference between the different concentration profiles. However, they are difficult to analyze. So when taking the second derivative of these curves, you get this nice profile, and then you're actually able to see the clear differences between the different concentrations in relation to the response on the sensor. And of course, this can be translated in a nice calibration curve, which of course you can use for interpreting the concentration in your measured sample. So we already heard a lot about our analytical validations, but also about our clinical validations. So for our bilateral validation, there are some additional tests you want to do before moving forward. You want to do recovery tests. Does my drug not stick to the filter? And we need to do some interference testing because we have no chromatography. We're just measuring everything at the same time in the same sample. And when we are moving to the clinical situation, of course, we want to compare our results with plasma. But also we want to compare the saliva samples measured with the two different machines. This will give you a little bit more information about how accurate the device is, also in relation to interferences of substances. We need to make sure that we are aware of the pH of the saliva in the patients participating in our trial to make sure, or to at least to understand whether pH is relevant for that particular compound. And also, we can have a look at the saliva flow. We can collect a saliva sample, which is just stimulated or non-stimulated. And of course, we could test pre-post meal effects. So these are the validation results of our levofloxacin assay. It has a nice calibration range covering the clinical range with a nice R-square. And when we look at LLOQ low, medium, and high levels, it all complies with the validation criteria. However, the interference testing is something interesting because you don't have separation of compounds in your sample, and you have to assess that effect of that other compound in your sample on your drug of interest. And of course, there are many other molecules that can interfere. And of course, they have to be in the saliva to interfere, and they have to have a UV spectrum, but of course, most drugs have a UV spectrum. So you need to make sure that you can handle that when you're analyzing a patient sample. So we did in this TB study, of course, a lot of interference tests with other TB drugs. So on the left side, you can see a gray curve, which was from clofazimine, and the black curve, which was levofloxacin. So the levofloxacin curve was significantly higher than the clofazimine curve. So in that case, there was no effect of the interference observed. On the right side, however, where there's pyrazinamide, the gray curve is higher than the levofloxacin. What happens in that case is that at the preferred wavelength for the measurements, you also measure a little bit of pyrazinamide. And that adds to your measurements, so your levofloxacin concentration has a positive bias. And of course, you want to make sure that you get rid of that bias, because otherwise you would make the wrong dose adjustments. So we looked at a lot of these different drugs that are used in TB or in co-infections like HIV. And for most of the drugs, there was no real issue. And of course, that's very convenient. And the wavelengths are specific for specific molecules. So that is actually a selective tool to make sure that you know interferences. There was only one drug, rifampicin, that had significant interference at lower concentrations. However, if we look at the TB treatment, levofloxacin is part of the drug-resistant TB regimen, and rifampicin is part of the drug-susceptible regimen, so you never give them at the same time. So although there is analytical interference, in real life you will not face that situation. So then we did a study in Tanzania that was supported by the Gates Foundation, and we looked at the value of looking at saliva concentrations in comparison to plasma concentrations. And due to feasibility issues, we were able to collect 2 samples, 1 and 4 hour post-dose, and we were able to use a Bayesian population PK models to collect AUCs. But I think the most important finding was that we were able to identify 8 of the 9 patients who were below target based on their, let's say, traditional plasma concentrations. So if we look at the impact of an assay, where there normally is no assay at all available, now suddenly you're able to detect patients that are likely to going to fail on their standard treatment. And we also looked at linezolid, also an important drug for drug-resistant TB. And if we look at these assay parameters like range, accuracy, precision, it also looks very nice. We see the same linear calibration curve based on our second derivative of the responses at the various concentrations. However, linezolid has significant more interferences. And that's simply caused by the fact that more drugs absorb at the same wavelength as linezolid. So we need to find a way to solve this problem. So this is the example of pyrazinamide at a concentration of 20 milligrams per liter, which is a, let's say, higher concentration for that drug, so around the peak concentration. And we thought, okay, linezolid as a drug concentration of 3 milligrams per liter, that is significantly affected. So in daily practice, what you could do is, instead of taking pyrazinamide first and linezolid afterwards, is take them at the same time. So pyrazinamide would be at that time only 5 milligrams per liter because it's a trough concentration. Hence, it does not interfere with your linezolid concentration. And so separating the drugs to avoid that interference is a practical approach to overcome these problems. There is, of course, a risk that you forget, but yeah, that's one of the main problems we often see in healthcare, that we forget something and that results in a mistake. So we need to be careful to make this work. But again, otherwise there's no assay available in some of these settings. So at this moment, we are running a trial in Vietnam, where we are sampling both levofloxacin and linezolid in 60 patients. This is a sub-study of the V-SMART study performed by Greg Fox and colleagues in Vietnam. And again, we're taking limited number of samples in again, plasma and saliva for validation purposes. And we are looking at efficacy and toxicity in this subgroup and see whether we can predict the same outcomes based on the saliva as well as on the traditional plasma samples. Of course, TB is not a big problem in many let's say, well-researched settings, especially also in Australia, but of course we see other drugs. Voriconazole is nowadays a frequently seen drug because of all the patients with cancer who are immunocompromised and need to be either on prophylaxis or on treatment. And of course, we well know that TDM is strongly recommended to manage voriconazole. So there has been done some studies looking at the relation between saliva and plasma. And you see the concentration curves on the left and the correlation curve on the right. And this all looks quite well. Of course, numbers are still limited, 10 patients. So what we subsequently did was develop a model based on this study, including the oral administration, plasma compartment, and saliva. And that model was able to predict plasma as well as saliva samples in a similar way. And in another study, they looked at what this would mean for real life. So if we were using plasma samples for TDM to improve target attainment, or we use saliva samples, you can see that the results for voriconazole using that model are fairly similar. So with one sample, you're already able to improve your target attainment with over 20 to 25%. An interesting discussion is, how do we classify whether saliva is suitable to replace plasma as a sampling matrix? So this review from 2016 had a crack at it and came with a kind of proposed classification. They say it's directly suitable in case there's no difference between your AUC in Cmax, Tmax, T half-life, etc. in both matrices. That makes sense. If everything is the same, you can replace it. On the other end, if the drug does not penetrate in saliva, it's not useful. Easy to understand. But the discussion is in the middle category. Likely suitable, likely unsuitable. In this situation, there are discrepancies in the parameters to a certain extent. One of the conclusions also from these authors were that sample sizes were small. There were mainly studies in healthy volunteers. There was large variability, and very few studies reported all the critical parameters for saliva collection, like the salivary flow and the pH. So some of these likely suitable, likely unsuitable can be resolved if we, for example, add information on saliva flow or on pH. I already know from the paper from 1978 that there are several anticonvulsants that can be easily measured in saliva, and they gave some recommendations for these drugs, and one was to measure compliance in pediatric patients. I think that's a rather strange conclusion because pediatric patients are not responsible for their own medication, so it's about the compliance of the parents of the patients. And I would rather use a different approach than measuring compliance, rather than supporting parents in making sure that target attainment is achieved in their children. Also, they said repeated sampling is easier with saliva. All makes sense. And if you look at the parameters for some of these drugs, and this one is inflammatory, the values are really spot on, same parameters, PK parameters for all these drugs. But the next step is, do we think that our pharmacists are capable of doing this in community? And that's another question. So when I reviewed literature, I came across a paper I actually co-authored myself. And in this large guideline on antifungal TDM, there was a very nice table on the knowledge, skills, and behaviors required to do TDM of antifungal drugs. And they were categorized as basic, intermediate, and advanced. And I think in a situation of TDM in the community, where a pharmacist supports a patient, I think we're looking at basic level. And if we see what basic level means, is that you are aware of antifungal toxicities and drug-drug interactions. And I think pharmacists are aware. They are aware of pharmacokinetics. They should be aware. Methods about adjusting the dose, we can support them with providing guidelines. If we go down, we see approach to timing of sampling can be put in a guideline. Again, we have the skills guideline-based dose adjustment. So just following the table, if we have this concentration, this is the dose, we need to do this. That all makes sense for community. And last but not least, we have provide the review and feedback to relevant clinicians. So indeed, I think pharmacists are very capable of reporting a result to a GP or a consultant in a hospital when they have measured a saliva sample. So I think that community pharmacists are perfectly suitable of doing this activity once they have properly been trained. So TDM and community pharmacy, I think we complement the existing TDM procedures available in hospitals and other settings with a semi-quantitative assay in community. It's easy to access and fast. You can just hand in your script and give a saliva sample. There are quite a few drugs that we can measure in saliva. And of course, it prevents additional visits, because if you can have easy access to a saliva assay to monitor, for example, your anti-epileptic medication, then you may detect at certain time when your level is declining over the next few scripts. And it gives you a easy tool to intervene rather than waiting for a convulsion to happen. And of course, we can expand with the tools we already discussed earlier, dry blood spot and VAMS, because a pharmacist can be a proper trained person. So in case there is an incidental collection of a sample in a patient, you're not able to teach the patient to collect that sample, but the pharmacist could and send it off to the relevant laboratory. So to conclude, I think point of care community is technically feasible, but we need to settle some criteria. So when do we consider the assay quantitative and when do we consider it semi-quantitative? And that is important. Otherwise, we, let's say, get rid of too many drugs that fall in the category semi-quantitative. We need to solve the amylase problem for interferences. We need to update the software on these devices, because the current software on these small mobile UV spectrophotometers is not suitable for TDM. But together with the School of Computer Science at University of Sydney, we have created a software package that could be implemented on such device. So there is option to do so. Of course, when we are providing as pharmacists a new service, there should be a kind of reimbursement for our time, for our disposables, etc. And of course, last but not least, we need a kind of regulatory approval process and accreditation for these types of assays. So I think the road is not paved yet, but at least there is a path forward. So I think, yes, we can. Thank you.